welcome to New York City Atheists live on tape, brought to you by New York City Atheists Incorporated. I'm your host, Dennis Horvitz, the David Letterman of the Hopelessly Damned. Several years, President Obama has lifted many of the bans on stem cell research done by federally supported laboratories, but not only has the United States, and it holds great promise for many diseases and conditions such as diabetes, Alzheimer's, and even heart disease. And it can be involved in rebuilding hearts. But not only have we fallen behind, but associated jobs are migrating to these countries. Regenerative medicine basically focuses on stem cell cures, and it holds great promise for many diseases and conditions such as diabetes, Alzheimer's, and even heart disease. And it can be involved in rebuilding hearts. But that's not the only, the migration of jobs is not the only problem. We have what so-called ethical considerations. There are many religious organizations that oppose the use of stem cells, probably in some cases because of very honestly held beliefs, but I'm sure in a lot of cases it's because of their specific religious agenda that they wish to impose on the general public. So one of the ethical considerations that I think we should be considering is the fact that there's a lag in science education in the United States. So here today to address this issue, possibly pose some possible solutions, is Dr. J. Peter Gergen, who is a professor in the Department of Biochemistry and Cell Biology and director of the Center for Developmental Genetics at Stony Brook University. And he has a list of credentials longer than my excuses for not having any. He is a graduate of the Massachusetts Institute of Technology. He has earned his PhD at Brandeis University. He did his postdoctoral work in developmental genetics under a Nobel Prize winner, and he told me how to pronounce his name, and I'm going to blow it. Eric Weishauss. Is that correct? No. He will correct me when Weishauss. Eric Weishauss, who is famous for his work on fruit flies. In any case, before I ruin this introduction any further, please welcome Dr. J. Peter Gergen.
Trying to get children, and then there would be leftovers. And so he got some parents to sign consent forms and said, okay, once we've done our assisted fertilization, we've got our children, we wanted to have 3, we've got all 3 of our kids. The ones that you fertilize and we don't need anymore, we're going to donate those to science, and you can use those to go and ask the human embryos have the same kind of cells as in the mouse system. So that was 1998. Because of the 17 years of work in the mouse system, it was pretty quick. He was able to show that they were there and that they had a lot of the same properties pretty quickly, in just a couple of years. And there was a rush. A lot of people started to say, okay, humans have these same cells. The potential is there to do a lot. A lot of people started doing a lot of things. And it really was going very, very fast. And that's when President Bush in 2001 just said, whoa, wait a minute. Where these cells are coming from is embryos that potentially have the ability to give rise to a human life that you're destroying to make these embryonic stem cells. We've got 80-some lines now. That's enough for the moment. Let's see what we can figure out with those 80 before we make any more. And so he had this edict to ban the use of federal money to generate any more human embryonic stem cell lines. At that point in time, in August of 2001, the first radio show I was on to talk about this, I actually agreed with him. I said, things are going really, really fast. We could use a cooling off period here of 6 months, a year, maybe a couple years to see what we've got our hands on and what's possible to do here. The thing that I don't want to get too political here or too philosophical, but it was sitting in that same position for 8 years without, he said, we're going to create a council, we'll get some people together that are really smart to talk about this and think about what we should do. Well, that council was never created. Nobody ever talked about what we should do. We basically sat in the same place for a long time. So that's up to 2001. Okay, so that's the history. We'll come back to the last 10 years and some of the recent advances in a few minutes. Before I do that, I want to talk a little bit about what the potential is here and get into some of the biology of actually talking about what a stem cell is, what do we mean by that. So the promise of stem cell research here, and this is a slide that's lifted from the director of the NIH in 2003, is that these cells, these embryonic stem cells, these cells that can grow forever and give rise to many, many different body parts, have the potential to treat lots of different human conditions. This is one of the things that I'd say people are looking to do is to treat these diseases. But these cells also provide the ability to study development in vitro, so as you watch these cells develop into different tissues, to see how that works. And these cells, since they're human cells, they also are the best place to go and ask about drug toxicity and drug specificity. We know that there are differences between human and mice. The FDA is not going to approve a drug based on a mouse passing clinical trials. It has to go through humans before it's given to the public. So there's a lot of promise there, and if we look at the numbers, the kinds of things that people think potentially could be treated with stem cell therapies is staggering. If you add this up, it's probably about 1 in 3 Americans that potentially have the ability to be treated for some condition. Heart disease is the #1 killer, and that's something that we think can be treated. I think it's mentioned in the press release that right now there's a clinical trial going on in Stony Brook where they're injecting stem cells into somebody that just had a heart attack, hoping that the stem cells migrate to the wound and are able to repair the heart. And so that's something that there's clinical trials ongoing now. Over the years that I've done this, I'd say there's 3 diseases that sort of stand out where the people that are the strongest proponents of stem cell research, and in particular thinking about the ethics of doing embryonic stem cell research and why it potentially is justified. And that would be diabetes, Alzheimer's, and Parkinson's, are the 3 that sort of stand out. Diabetes, we known for a long time exactly what the problem is. It's the beta islet cells in the pancreas are dying. You can't metabolize insulin properly. If you could replace that cell type, then you could cure diabetes. And actually one of the leading researchers in stem cell science in America is a researcher at Harvard who has got a lot of money from the Juvenile Diabetes Foundation to study exactly that process. And his interest in doing that, he used to also work on amphibian embryos, is he's got a kid with diabetes. And once he saw the potential, he said, okay, my lab is now going to change what it's working on, and we're going to try to cure diabetes. The other 2, Alzheimer's and Parkinson's, both of those diseases we know are due to neurodegeneration. Cells in your nervous system that are dying, that the nervous system starts to become dysfunctional. In one case, you forget where your car is or how you got here, which hopefully I don't get Alzheimer's during this talk today. Parkinson's, you lose motor control and you start to shake. Both of those tend to be late onset diseases, but if someone in your family gets that, it's very debilitating, not just to that person, but to the whole family structure in terms of the impact on that person. I could mention that one of the leading proponents of embryonic stem cell research was Nancy Reagan, after Ronald came down with a neurodegenerative disease. And so she sort of changed colors a little bit there based on her own personal condition. And for all 3 of these diseases, now there's good reasons to think that yes, we are going to be able to treat these with stem cell research. People have made mouse models of all 3 diseases, and using stem cell-based therapies, you can start to ameliorate or cure the condition. And there's some proof of principle experiments with human cells showing that we're not that far away from potentially being able to do that. Okay, so that's why everybody cares about this so much, is that the impact on human biology and human medicine is quite significant. So let's step back and just ask, what is a stem cell? So a cell, for those of us that are multicellular organisms, this is one of the building blocks. It's one of the fundamental building blocks of our body. The other word here is the word stem, and when I went to give this talk the first time, I said, okay, let me go look up stem in the context of cell and see what it says. Wikipedia didn't exist then, so it wasn't possible for me to go there and look it up. But there's 2 sorts of definitions you see in Webster's. One is the main upward growing axis of a plant, so a long stem rose is a really good thing we're carrying around today. And the other is the main line of descent of a family, and I think this is really where the word stem cell comes from for the biologist. So what a stem cell means to a biologist is depicted on the next slide. This is the basic definition. In my undergraduate course where I teach this is the one definition. If they can't write this down at the end of the semester, then they're not going to pass my course. That there's the basic definition of a stem cell. It's a cell that's going to divide and produce two daughter cells. So our cells divide by fission, one cell produces two, where at least one of the two daughter cells retains exactly the same properties as the mother cell, with the key property being the ability to divide and produce two daughter cells. So that's the definition of a stem cell. The ones that are in the news are the embryonic stem cells. And the early embryonic stem cells, the one green cell divides to give rise to two green cells. And so you get this expansion of the stem cell population. But at later stages of development, we have stem cells maybe in the nervous system or in the blood system, the hematopoietic system, where a cell will divide, it's a stem cell, it produces one daughter stem cell, but then it produces another cell that has a different phenotype that will start to differentiate and produce different types of structures. This is clearly, for multicellular organisms like ourselves, the ability to proliferate like this, to retain cells, you have the ability to divide and produce more cells is a key developmental strategy. So I want to talk about, we'll get back to embryonic stem cells in a minute, a couple of common examples of other stem cells that are in our body that we've known about for a long time. One of them, and in this, the juxtaposition of these two, I'll also sort of point out what are some of the key issues that stem cell researchers are facing in terms of trying to harness the power of these cells. So we all know about hematopoietic stem cells. These are cells that normally reside in your bone marrow, and they have the capacity to generate all the different cell types. Like if somebody has leukemia right now, one of the standard ways to treat it is you basically kill all of their leukemic hematopoietic cells. You give them a bone marrow transplant with a donor that's sufficiently well matched, and you recreate their immune system and their hematopoietic system using the stem cells that were in the donor's bone marrow. So hematopoietic stem cells, we know quite a lot about them. We have molecular markers that you could go to the bone marrow and you can actually purify cells and say, here's a population of hematopoietic stem cells that is pure, that I know exactly what genes they're expressing and what they can do and what they can't do. One of the big problems with hematopoietic stem cells, even though we know exactly who they are, we can't grow them in vitro. You think about it, if we could take hematopoietic stem cells out of somebody, out of their bone marrow, and put them in a test tube, and could recreate the environment of the bone marrow, you wouldn't need blood drives anymore. You wouldn't, like on NPR the last week, they've been advertising that the snow sort of knocked out blood drive collections for about a week and we're falling behind. That wouldn't be an issue. If you could grow hematopoietic stem cells out of bone marrow, then you wouldn't need to do blood drives anymore. You'd create a bank of the different sort of diversity of hematopoietic stem cells you need to potentially give people blood transfusions, and whenever you needed some, you just take some out of the freezer and grow them up. That we can't do. And so that's a big issue. And that's, in terms of developmental biology, a lot of what's being done with different types of hematopoietic stem cells is try to understand the environment that they normally grow in so that we can recapitulate that, we can reproduce that in vitro. Okay, so this is just a cartoon of a hematopoietic stem cell. This is a self-renewing cell that gives rise to cells that are slightly different. These also can be stem cells. We can have myeloid or lymphoid stem cells that will continue to produce daughter cells that retain that sort of intermediate differentiation. But at the end of the day, in the hematopoietic system, we produce lots and lots of different cell types. Okay, so let me talk about a second example, and we'll see here a difference in terms of what we know and what we can do. It's the stem cells in your skin, dermal stem cells. So we know we have them there because if you cut yourself, or let's say you skateboarding and you go sliding across the pavement and you mess up a bunch of your skin, your body will fix itself. That the stem cells there will get called to arms, will produce more cells, and those will differentiate and replace your skin. The skin that's on your surface now, those cells are already all dead, and they're being replaced constantly from the stem cells that are underneath. What we know in this case is that you can go to somebody's leg and you can do a biopsy, take out a little plug of the dermis about the size of a dime. And in this case, we know the culture conditions for growing those cells, that you can take that out, put it in a culture dish, and in 2 weeks you can grow enough skin in vitro to cover the entire human body. So this little plug can produce enough cells that are doing the right thing. And that's the person who I learned this from, is the director of the Living Skin Bank at Stony Brook. The major job of this skin bank, when a burn victim comes into the hospital, is that the doctors in the emergency room need to keep that guy alive for 2 weeks until they can grow the skin to give the guy the skin transplants and put it back on. And so in this case, we know how to culture the cells. We can grow them, but in terms of the stem cell biology and understanding who's doing what, within this plug of tissue that you're taking out, there's still controversy as to which is the stem cell. So we don't understand the biology well enough to really know which is the player that's doing this. So we know now in adults that there are stem cells in basically all the major body organs. And if we think about it for a minute, that probably has to be true. You live now for maybe 80, 90, we're approaching where more and more people are living to 100, that tissues, cells in all of your tissues are dying off with time and they need to be replaced. And if they're not replaced, then you're going to lose that organ. And what's happened over the last 20 years is people going to different organs and finding that there were different types of stem cells. Back in 2001, everybody thought, well, the one place we know there's no stem cells is the nervous system. Because if you break your spinal cord, it's not going to be repaired. Those cells don't exist there. But we know now that there are neural stem cells. And actually, there are neural stem cells in the peripheral nervous system as well as in the brain. And these cells can give rise to lots of things. And the trick now, with spinal cord regeneration, there's clinical trials ongoing where they think it's actually possible to do this. And the key is that you need to be able to get the cells there quick enough. You have a couple of days after you've severed things where the environment is such that it's possible to rebuild things. So there are stem cells in all these different tissues. Okay, so let's return now to stem cells from embryos. I'll give you sort of an overview of all we need to know about mammalian embryology with respect to stem cells is really on this slide here. Sperm and egg get together, fertilization, we make a zygote. This zygote, so this single cell that's now diploid, it's got DNA from both mom and dad, is going to go through several rounds of cleavage, division, where it's going to produce daughter cells that are identical. We've gone here from 1 cell to 4 cells, we've gone through 2 rounds of division. What we know is that these first couple divisions, that all of these cells are, a word I'm going to use is totipotent, that they're capable of making an entire human. And we know that because you get identical twins. Where you get an identical twin is this 1 cell divides, you get 2, and then somehow they get separated, and they give rise to two individuals inside the womb. So we go through about 6 divisions, 5 to 6 divisions, where the cells are small round balls and they're all the same, and they're sort of in this growing clump of grapes inside that's developing. And then when you get out to the 64 to 128 cell stage, you get your first differentiation. And that's the cells that are on the inside stay as little round grapes. The ones that can sense that they're on the outside, that they're sort of being pushed out by the others, will flatten out and make a tissue that's called the trophectoderm, a thin layer of cells that's basically a protective sheet around these inner cells. These inner cells have the really creative name of inner cell mass. So the inside cells is the clump of them. That's the inner cell mass. And the stuff around the outside is called the trophectoderm. These stages here, to come back to in vitro fertilization and doing development in a test tube, all of these steps we can do in a test tube. And the people that do assisted fertilization, reproductive technology now, they'll watch the embryos develop up to the blastocyst stage and then try to pick out the ones that look the healthiest to implant back into the mother. After this stage, so the blastocyst, this is at about 5 days of development, wants to implant into the uterine wall. From that stage on, we can't do it in a test tube. We haven't figured out how to grow a uterus in a test tube yet to get the embryo to implant. This we can do in vitro. After this, you need to have a female. Okay. This will just sort of, same pictures now in color, a little more recent picture. Zygote, we get out to this blastocyst stage. One of the points I wanted to make here as we go from zygote to blastocyst now to an embryo that's implanted, is that as here all the cells are the same, here we've got 2 different cell types, the inner cell mass, and then the cells are on the outside. When we get a little bit later in development, the cells become more and more differentiated. They become more and more specified in terms of what they're expressing and what they're going to do. And one of the things that correlates with that, so the zygote, the cells are totipotential, they can give rise to anything. These cells here, the embryonic stem cells, they're pluripotent. They can give rise to lots of different cell types, but they're not totipotent. You can't get a whole animal out of one of these embryonic stem cells. And then as you go down to the different germ layers, the gut, the muscle, and the skin, the cells become more and more restricted. Their normal potential, what they're only going to give rise to, becomes more restricted. So these cells, the ones in the blastocyst, these are the culprits, the embryonic stem cells, where you can take these inner cell mass cells out, you can culture them in vitro, and then if you change the cocktail of growth factors and cytokines they're being exposed to, you can get them to differentiate into all these different pathways. This is the stuff that people were doing for 17 years with mouse embryos. That James Thompson said, if human embryos have these same kinds of cells, we should be studying these cells to figure out what they might be useful for.
was nobody served.
Okay, so some of the lessons from the mouse, I already went over these. So these embryonic stem cells, or ES cells, they can be cultured in their pluripotent state. We can induce them to differentiate. We can modify them genetically by putting DNA, engineer them genetically by putting DNA back in. And in the mouse system, you can actually take these cells, you can put them back into a blastocyst stage embryo, and if one of those cells ends up in the germline, you can get, in the next generation, a mouse that came from the cell that you've genetically manipulated. Okay, so human stem cells, some of the issues, embryonic versus adult. The embryonic stem cells, obviously, they're isolated from embryos. The adult stem cells, isolated from adults. Here we know exactly where to go. You go to the blastocyst stage, you take out the cell mass. Here it depends on the tissue. In some cases we know exactly who they are, in some cases we don't. Embryonic stem cells, easy to grow, relatively easier to grow and manipulate. The adult stem cells, in some cases, so the dermal stem cells, we know how to grow them, we can't purify them. Hematopoietic stem cells, we can't grow. The embryonic stem cells, we believe or think there's a lot of evidence that you can generate many, if not all, different cell types using these. The adult stem cells, it's not clear that you can do everything, although at the last couple slides I'll show you some of the recent developments that say that stem cells from adults may be very, very useful. One of the issues with embryonic stem cells in terms of therapies is that if you're going to, just like with a bone marrow transplant, you need to find a person that's a good match so that you don't end up with a rejection issue. That if you're going to do regenerative medicine, you're going to replace somebody's cardiac tissue or some of their pancreatic tissue with stem cells. Those stem cells need to be a good match. And that was one of the people that wanted to do this kind of medicine, the doctors, one of their major objections to Bush's edict, so we've got 80 lines, that's enough, is that those 80 lines weren't very diverse. I think they were from about 6 different labs, not representing the diversity that is in the human population across the earth that was not going to be useful in the long run for doing therapies. With adult stem cells, the very potentially really important thing here is that if you could take a cell out of me and then use that to grow some pancreas or some liver or some heart, there's no transplant problems. You could then go back and repair me using the cells that comes from myself. Okay, so I want to talk now a little bit about, so embryonic stem cells, there was a lot of reason to think that they had a lot of potential and they were very useful. There's also this big ethical issue that you're potentially taking a life away. Here's a blastocyst stage embryo that could possibly become a human being that you're sacrificing. So the traditional derivation of human embryonic stem cells would go to this blastocyst stage embryo, remove the inner cell mast cells and make an embryonic stem cell line. The traditional way of doing that, when you isolated these cells, you were destroying the rest of the embryo. One of the things that people played with for several years was coming into these blastocyst stage embryos, and let's take out a single cell and leave the rest of the embryo intact. And people got pretty good at that. It's actually, there's a couple of papers where people have shown that they could take a cell out, create an embryonic stem cell line, and that the rest of the embryo, if you implanted it back into the mouse, you could get a mouse pup. So it was possible to do the surgery, and that was one way around, technically, around the ethical problems. There's still an ethical problem, unless your batting average is 100%, then people would still say, okay, you're still at some level destroying embryos, and so this is not acceptable. What we'll call as a sort of curveball around this is something called parthenogenesis, which I won't get into the details of this, but basically we're taking an egg and not fertilizing it. We're not making a diploid individual that's got DNA from mom and dad. We're doing parthenogenic activation, getting some cells that are diploid, where they've got both copies of their DNA from their mother, that can make it to the blastocyst stage. So they can make one of these guys, and you can get the inner cell mast cells out and make an embryonic stem cell line. But we know that these parthenogenetically activated embryos, pseudo-embryos, are not capable of going to term. So here you haven't killed anything that could grow up into be a human being. An alternative is, and this is where it's a process that's called therapeutic cloning by a lot of people, is an alternative to embryonic stem cells. It involves a technique called somatic cell nuclear transplantation. So what's shown here, this is a picture of a mouse oocyte, where what we're going to do is we're going to go into that mouse oocyte where there's a haploid nucleus that the mom put there. We're going to take a needle and we're going to take that nucleus out. And then we're going to replace the genetic material that's inside of that egg, the cell that is ready to undergo embryonic development. We're going to put in DNA that comes from an adult cell. Okay. And so that's this somatic cell nuclear transplantation cartoon here. The egg or the oocyte, we take the nucleus out, Then we get a nucleus from a somatic cell, let's say a skin cell or a fibroblast, get that to be introduced into the egg, and then trick this egg into thinking that it should start development. It goes out and makes a blastocyst stage. You can take these cells out, these intercell mast cells that are diploid, they have DNA that came from the donor somatic cell, whatever it is, and make an embryonic, not an embryonic stem cell line, but a pluripotent stem cell line that potentially is useful. If we were to carry this on, if we were to take this blastocyst, where the DNA came from a donor somatic cell, and this slide, just in terms of the legal issues, there's different places, and we'll go right to the end here, that depending upon who you are, some what we'll call more conservative politicians would say that we shouldn't even do this, that transferring DNA in from a somatic cell into an egg, that is the prelude to cloning, right, going all the way to term here. And so we shouldn't even allow that. Some of the names here you might recognize a little bit more liberal bent, were to say, okay, this is all good. We can go down here, we can make these pluripotent stem cells, these things that behave like embryonic stem cells, and use these for regenerative medicine. This is exactly what we call therapeutic cloning. And what these guys said is it's okay to do all of this, but we're just going to not allow people to do the cloning thing.